Okay, hello everybody. Welcome to Chapter 19. We're going to be dealing today with the composition of blood. So this will be the second installment in what will be a growing familiarity with the first installment in what will be a growing familiarity with the cardiovascular system. And we should note just for a minute what the cardiovascular system is for. Early in evolution, life was relatively simple. Unicellular organisms essentially swam in the environment from which they procured their food and secreted their waste products directly across their cell membranes without a lot of specialized structures or adaptations. But as the environment on the planet changed, it necessitated a switch in the type of organisms that populated the planet in order for them to survive these changing conditions. So we went from unicellular organisms to teams of organisms acting as biofilms that had certain specialized communities in the biofilm good at certain tasks, such as defending against invaders or attaching the community to a substratum or procuring food. And over time, those biofilms had among them cells that became so interdependent that they couldn't live without each other, and that was thought to be the development of the first tissues. From there, we moved to organ systems and very complex organisms. As the body plan became more complex, cells in the body became distantly removed from where the oxygen and the nutrients come in. And so we have to come up with a way to delivering those vital components to the cells in order for the entire organism to survive. And that really is thought to be the reason that the circulatory system evolved. You could think of it as the body's pizza delivery and trash removal service, among other things. So let's talk a little bit about the blood, which is what flows through the vessels of the cardiovascular system. Just a thumbnail sketch. The cardiovascular system consists of a blood pump, the heart, the blood vessels, which carry blood to the cells of the body and back from the cells of the body. And it works in concert with the respiratory system and the digestive system. The respiratory system supplies oxygen to the blood and removes carbon dioxide. And at the same time, the digestive system provides nutrients to the blood and removes some waste products. So all these systems work together in order to deliver oxygen and nutrients and remove carbon dioxide and waste products from the cells and keep them happy and humming along. So the blood pump, the heart, the blood vessels, and the blood that carries those components around the body are essentially what we think of when we think of the cardiovascular system. Today we're going to be looking in depth at blood. What does blood do? Well, it transports gases, nutrients, and waste products around the body, drops off oxygen and nutrients to metabolically active tissue, and removes the waste gas carbon dioxide and breakdown products, waste products, such as urea and ammonia and other breakdown products, many of which contain nitrogen, from metabolically active tissue. If these waste products aren't removed, what will happen is that the chemistry around these tissues and cells will change to the point where the cells and the tissues will no longer function. What else does the blood do? It transports processed molecules around the body. As an example, the precursor of vitamin D runs from the skin to the liver and then to the kidneys. Active vitamin D is important in mineralizing bone. It transports regulatory molecules such as hormones. We discussed at length how the endocrine system operates in Chapter 18, and we noted that this chemical signaling system relies on the circulatory system as a transport vehicle to carry these chemical messages around the body. And it also regulates pH and osmosis, so controls electrolyte balance and fluid balance in the body. Normal pH of most body tissues is around 7.3 to 7.45, and the blood has no small part in keeping the pH within that relatively narrow range. It also maintains our body temperature. Our warm blood is shunted to the interior of the body to keep our vital organs operating at the same time. When the body heats up too much, the blood is shunted to the surface so that it can cool as it comes into contact with tissues at the periphery, such as your skin. On a hot day, you already know that you sweat. The sweat, as it dries, cools the skin, which cools the blood, which in turn cools the body back down to normal temperatures. And on the flip side, when it's cold, the blood gets shunted to the internal organs in an effort to keep them operating, while not concerning ourselves so much with your skin temperature at that point. So maintenance of body temperature, very vital, right? We can't get too cold or too hot, because in either instance, chemical reactions that keep the body going would stop. If we were to get too warm, the enzymes that allow chemical reactions to happen would denature, and those chemical reactions would cease. Not to mention structural proteins would begin to denature. If we were to get too cold, there simply wouldn't be enough molecular movement for chemical reactions to happen at a rate that would sustain life. It also provides protection against foreign substances. Example of that is the antibody molecule, which you could think of as the magic bullet that destroys a particular pathogen by tagging it for destruction, either through chemical means, such as what we'll learn about in the lymphatic system chapter, the complement system, or by attracting macrophages to the area to destroy the tagged either infected cell or pathogen. And then finally, it's important in clot formation, which is a vital part of hemostasis. When you get a puncture or a laceration, you bleed, but you don't continue to bleed. Eventually what happens is that a blood clot will form, and that clot will form a temporary plug in the injury and allow you to hold on to this vital fluid, which we know is blood, and of course all the vital components that are in the blood. If we failed to do this, if we didn't make a clot, we would bleed out, and the loss of blood would mean a loss of oxygen and nutrients, and that would eventually result in a shutdown in body systems. So what you're looking at here is what happens to whole blood when we take a small sample of it, put it in a centrifuge tube, and spin it down. And what you find is that you generate three distinct layers. You generate a packed red cell layer, which we define as essentially the hematocrit, the percentage of red blood cells that make up the total volume of blood. Average individual, it's about 45%. And we have a buffy coat, which is primarily platelets and white blood cells. The platelets are the agents of blood clotting, and the white blood cells defend against disease. And the rest is this straw-colored liquid called plasma, which contains mostly water and ions, but also waste products, proteins, hormones, and molecules that are designed to carry signals throughout the body. We've already talked about hormones in the endocrine chapter. We should also note that there are defense proteins in the plasma, such as the proteins that make up complement, and immunoglobulins, which defend against disease as well. And there are also proteins that are designed to allow the blood to maintain its volume. And the most important example of that is blood albumin, which is essentially an osmotically active large protein molecule that draws water into the circulatory system from surrounding tissues. And if we break down the components of blood, we can look here at the plasma, right? You can see about 7% protein, 91% water, and then there's other solutes in there. And if we look here at the hematocrit, it's mostly erythrocytes, okay? The erythrocytes are the oxygen carriers. They are anucleate disc-shaped cells filled with hemoglobin and carbonic anhydrase, and their job is essentially to escort oxygen around the body from the respiratory system ultimately to metabolically active tissue, at which point they deliver their oxygen payload and pick up a small amount of carbon dioxide. Most of the carbon dioxide that's carried away from active tissue is actually carried in the plasma. If we look at the buffy coat, we find white blood cells, and we also find platelets. And if we look at the white blood cells, we see that there are about 60% neutrophils, about 25% lymphocytes, about 3 to 8% monocytes, 3 to 4% eosinophils, and then the basophils bring up the rear at about 1%. And what do the white blood cells do? They defend us against disease. Okay, there's an easy way to remember the order of abundance of the different white blood cells is the mnemonic "Never let monkeys eat bananas." That's from the most frequently encountered white blood cell, the neutrophil, all the way down to the least frequently encountered white blood cell, which is the basophil. The platelets, of course, are essentially bags of clotting factors. Their job is to promote clot formation in the event of an injury. You can see here the breakdown in the plasma, right? We've already talked about the fact that there's ions, nutrients, waste products. There's a lot of carbon dioxide in plasma, as well as a significant amount of nitrogen gas. And of course, there are hormones, chemical signals that the body uses to coordinate homeostasis. The proteins, we've talked about what the albumins do and the globulins. The fibrinogen is one of the components of the clotting cascade, which is basically a series of protein factors in the blood which, when triggered as a result of injury or infection, will generate an extracellular cascade that will eventually result in the production of a fibrin clot, and that's what will keep us from bleeding to death. So let's look at the plasma. It's the liquid part of blood. It's a colloid, so it's a liquid that has suspended substances that don't settle out of solution. It's 91% water, the rest being proteins, ions, and nutrients, as well as waste products, gases, and regulatory substances. The proteins include the albumins, which are there to maintain the osmolarity of blood, provides viscosity. It's also a buffer, as are most proteins. It transports fatty acids and bilirubin, which is a breakdown product of hemoglobin, and also thyroid hormone. The globulins are lipid transporters. They also are going to interact with antigens, which are ID tags on cell surfaces that either distinguish that cell as belonging to the body or as being pathogenic. Okay, the antibodies are there to recognize that. They tolerate our own antigens and they attack the antigens of pathogens. Complement is a series of proteins that is activated in the event that an antibody recognizes a pathogen and binds to it. The complement cascade will result in the destruction of the infected cell or the pathogen as a result of producing chemical reactions that either punch holes in the infected cell or pathogen or cause that infected cell to commit suicide in a process called apoptosis. The fibrinogen is the soluble precursor of the fibrin clot, so that's also a major plasma protein. Essentially, the fibrinogen is waiting around for a clotting cascade to take place. It's converted from the soluble fibrinogen to fibrin by an enzyme called thrombin, and that results now in the insoluble protein clot that keeps you from bleeding out. Okay, so those are the major proteins in the plasma. Most of these are made in the liver, and this is one of the reasons why when the liver begins to fail, we begin to have problems maintaining blood pressure and blood volume, as well as problems with fluid balance throughout the body. In addition to that, we become more susceptible to disease because we don't have as much of these defense proteins in the plasma. Again, the liver being the major place that these things are produced. Okay, the plasma contains ions. They're involved in osmosis and in the maintenance of membrane potential and in acid-base balance. Major ions include sodium, potassium, and calcium, among others. Nutrients are also found in the plasma. These include glucose, amino acids, triacylglycerols, cholesterol, and vitamins. And then there's waste products. Most of these contain nitrogen. Examples of nitrogenous waste include urea, uric acid, creatinine, and ammonia salts. They are the result, essentially, of protein breakdown. We don't want these building up in the body because they will radically change the pH of the tissues and the tissue fluid in the plasma. So we have to control their production and their secretion, their excretion from the body. Bilirubin is a breakdown product of hemoglobin, and lactic acid is an end product of anaerobic respiration. The gases that are transported in plasma, some oxygen, mostly carbon dioxide, and lots of nitrogen. Regulatory substances that you find in the plasma include hormones and enzymes. We've talked about hormones in the endocrine chapter. Remember that enzymes are there to promote chemical reactions so that they can take place at a rate that assists the cell in its survival, essentially by lowering the activation energy that blocks many chemical reactions going from reactants to products. This is just a breakdown of the composition of plasma. This is 19.1 in your book, so take special note of the information here. It could very easily show up on an exam or a PRS quiz. Let's talk a bit about the formed elements. Okay, the formed elements is just a catch-all term for the cells that you find in blood. Okay, so these are the platelets, the red blood cells, and the white blood cells. The red blood cells are erythrocytes. The prefix erythro means red. Are biconcave anucleate discs that have lots of hemoglobin and an enzyme called carbonic anhydrase. And their main job is to transport oxygen and a small amount of carbon dioxide. The white blood cells are the defenders against disease, and they can be broken down into two broad groups, the granulocytes and the agranulocytes. The granulocytes have visibly staining granules in their cytoplasm. Examples of the granulocytes are the neutrophils, the eosinophils, and the basophils. The reason we make a big deal about these granules is because they contain chemical components that help fight disease. Essentially, you could think of the granules as the body's version of mace. In the event that we encounter a pathogen, a bad actor, these cells will degranulate. They'll spray their chemical contents on the attacker, and this will set into motion a series of chemical reactions that will ultimately destroy it. The agranulocytes have granules, but they don't stain. Examples of agranulocytes include the monocytes, the macrophages, and of course the lymphocytes. And these are very important components of our specific defenses against disease. You could think of the granulocytes as our nonspecific barrier against massive infection. They use the same chemical attack regardless of the identity of the pathogen, whereas the agranulocytes tailor their chemical attack to a specific strain of bacteria or virus or parasite or what have you. The lymphocytes, probably the most important part of this collection of cells. The macrophages are important because they digest pathogens as they come into the body, break them up into little pieces, and display those pieces on the membrane surface as a warning to the lymphocytes, which when they recognize a particular antigen will be stimulated to divide, generate an army that will then go out, attack, and destroy the invading pathogen. The platelets are cell fragments that assist in blood clotting. They form platelet plugs and they release the chemicals necessary to produce the fibrin clot. Hematopoiesis is the process of blood cell production. The stem cells are formed elements predecessors. Essentially what happens is that in the marrow, the red marrow, these stem cells as a result of interaction with particular chemical signals, will begin to change their appearance and their behavior. So they'll go from what we call an undifferentiated to a differentiated state. As a result, they are the progenitors of all the formed elements, the white blood cells, the red blood cells, and also the platelets. Proerythroblasts will become red blood cells eventually. Myeloblasts will become one of the granulocytes, and the lymphoblasts will become the lymphocytes. The monoblasts will become monocytes, which in turn will become macrophages. The megakaryoblast are essentially platelet producers. So the small fragments of the megakaryoblast break off and become now these tiny membrane enclosed sacs of clotting factors that are one of the more abundant elements in the plasma. Okay, remember, all of these cells are suspended in the plasma, and remember that blood itself is a connective tissue because of the large amount of extracellular matrix and the relatively small amount of cells compared to the volume of plasma. So it's one of the two liquid connective tissues in the body, the other one being lymph. This is just a rundown of the formed elements of blood. This is 19.2 in your book. We'll go through each of these in detail in just a bit. But what I wanted to show you here was essentially the heritage, if you will, of the formed elements. We start initially with a stem cell, a hemocytoblast, and these are found primarily in the red bone marrow. They will differentiate into either myeloid or lymphoid cell that are going to produce now essentially all the rest of the formed elements of blood. The lymphoid line is going to develop into lymphoblasts, which will eventually produce the lymphocytes. The lymphocytes come in two flavors: the B lymphocytes, which are the antibody producers, and the T lymphocytes, which do a whole lot of things. Among other things, they actually show up at the site of diseased tissue and physically destroy it. And they also show up where pathogens are found and engulf and destroy them and chemically attack them. Those are called the cytotoxic T cells. The regulatory T cells serve as an off switch in order to turn down the immune response after it's performed its task. And then we have the helper T cell, which facilitates the production of armies of B and cytotoxic T cells that go out and are basically crafted to destroy one particular strain of pathogen. Okay, so that's the skinny on the lymphocytes. If we go over to the myeloid line, we find proerythroblasts, megakaryoblast, and myeloblasts. Let's go with the myeloblasts first. The myeloblasts form progranulocytes, which then give rise to eventually the granulocyte population, the basophils, the eosinophils, the neutrophils, and these are part of our body's non-specific defenses, okay, the granulocytes. The monoblasts will eventually become monocytes, which will eventually become macrophages. The job of a macrophage is to simply patrol the body and engulf and destroy any pathogen that it finds or any unusual molecule that it finds, break that pathogen or molecule up into tiny pieces, and display portions of it on the cell membrane as a warning to the lymphocytes that they need to generate now an army of cells specifically designed to destroy that pathogen. Okay, so that's essentially why we need the monocytes and the lymphocytes working as a team. Really, one can't perform well without the other. You could think of the macrophages as one of a family of cells that we call antigen-presenting cells. There are many cells in the body that have the ability to engulf pathogens and display portions of those pathogens on their surface. They're called APCs. The macrophage is an example of an antigen-presenting cell. If we go over here, the megakaryoblast precursor is the essentially going to produce the platelets. Okay, the precursor to the megakaryocyte generates the megakaryocyte, which then simply sits in blood sinusoids in the red marrow, and then tiny fragments of the megakaryocyte break off and they form the platelets. The platelets are anucleate cell fragments that contain clotting factors. The reason that this cell is called a megakaryocyte is, number one, it's big. That's where the mega comes from. Karyo, K-A-R-Y-O, indicates nucleus, and cyte indicates cell. So this is a cell with a big nucleus. And it's unusual in the sense that it's one of the few cells in the body that undergoes mitosis without cytokinesis, and this accounts for the large nucleus. The proerythroblast eventually becomes the early erythroblast, and these eventually become reticulocytes that lead to red blood cells. Red blood cells are anucleate oxygen carriers filled with hemoglobin, which is the oxygen-carrying pigment, and carbonic anhydrase, which aids in the transport of carbon dioxide, but also more importantly, helps to maintain the pH balance in the body fluids. If we look at the red blood cell in a little bit more detail, we find that it's a biconcave disc. So it looks a little bit like a frisbee or maybe two dinner plates glued together. Okay, and the reason for this is that it increases the surface area, which is very important to the function of this cell, the main function of the cell being diffusion and gas transport. So the more surface area we have, the more diffusion can take place. It's found in higher concentrations in male than female plasma, and the reason for that's very simple. That's the effect of testosterone. It raises the hematocrit, and the reason for that is that testosterone also increases bone density and muscle mass. In order to do that, you have to have more oxygen carrying capacity in the blood because muscle and bone are metabolically active tissue. It's about a third hemoglobin and then a third lipids, ATP, and carbonic anhydrase. The ATP, of course, is the energy source, and the lipids form the components of the cell membrane. Okay, so that's a lot of hemoglobin for one little cell to carry around, and that's also why it lacks most of the other organelles, because they would take up valuable space inside the red blood cell that could otherwise be occupied by the all-important hemoglobin. Because they lack a nucleus, they lack biosynthetic capability, and they can only make a couple of trips on average around the circulatory system before they have to be recycled and those breakdown products reused to make new red blood cells. Their main function is transport. They take oxygen from the lungs to the tissues. 98.5% of this is attached to the hemoglobin in the RBCs, and a little under 2% is dissolved in the plasma. It also takes carbon dioxide from the tissues to the lungs. 7% is dissolved in the plasma, 23% in the hemoglobin, and 70% is transported as bicarbonate, generated as a result of a combination of water and carbon dioxide and the action of the enzyme that catalyzes that reaction, carbonic anhydrase. Okay, so where are these bicarbonate ions? They're in the plasma. They're also found in the red blood cells. Okay, most of the carbon dioxide is not found bound to the hemoglobin. But that's not to say that the CO2 that's bound to the hemoglobin isn't critically important. It is. As we're going to find out, there's sort of a push-me-pull-you relationship with oxygen and carbon dioxide interacting with hemoglobin, in the sense that when the red blood cells are in a high oxygen environment, they tend to pick up oxygen and exclude their bound carbon dioxide, which then eventually gets eliminated from the body when we exhale. And on the flip side, when carbon dioxide is in high concentration, the hemoglobin tends to cough up its oxygen and deliver it to metabolically active tissue, while the CO2 tends to bind the hemoglobin and get carried away so that it can eventually be eliminated from the body. All right, so you can think of hemoglobin as a flexible transport molecule that is going to respond to changes in the concentration of these gases in a way that enhances the transport of oxygen to metabolically active tissue and the transport of carbon dioxide away from it. Hemoglobin is a combination of protein and a cofactor called heme. Heme is this really funky looking organic molecule that has an iron atom at its center surrounded by pyrrolidine nitrogens. The iron is what binds to the oxygen molecule. So you can see that in every hemoglobin molecule you've got 4 polypeptide chains, 2 betas and 2 alphas, and they each carry a single heme, each of which can carry an oxygen molecule. The types of hemoglobin that you see in human systems include embryonic or fetal hemoglobin, which has a greater affinity for oxygen than adult hemoglobin. The purpose of that, again, is to steal oxygen from the mother's blood supply while the baby is developing as a result of diffusion and binding and transport. The embryonic or fetal hemoglobin turns off shortly after we're born, and it's followed by the expression of the adult hemoglobins, alpha and beta. Okay, in the adult, we have different forms of hemoglobin. The normal forms are alpha and beta. There are also disease forms of hemoglobin, such as sickle hemoglobin, which is less capable of carrying oxygen at low oxygen concentrations, and it is the agent of sickle cell anemia. Okay, so you've got the embryonic hemoglobin, you've got the adult hemoglobin. Hemoglobin is termed oxyhemoglobin when it's saturated with oxygen, and it's termed deoxyhemoglobin when it is bound primarily to carbon dioxide. And remember, I told you guys that the oxygen and the carbon dioxide sort of fight for space on the hemoglobin molecule. It's an allosteric interaction, however. The binding sites for the carbon dioxide are physically different locations on the hemoglobin molecule than the oxygen binding site, which of course is square in the middle of the heme. Okay, but they do affect each other. As oxygen tries to move into hemoglobin, carbon dioxide gets pushed out, and on the flip side, as carbon dioxide tries to move into hemoglobin, it squeezes the oxygen out. So let's take a look at hemoglobin and oxygen transport.
All cells need oxygen. It is the essential fuel which is necessary to enable cells to stay alive and to carry out their various activities. Bringing oxygen to the cells requires the uptake of oxygen from the air in the lungs, its transportation in the blood, and its delivery to cells all over the body. The first step is the taking up of oxygen by blood flowing through fine capillaries in the walls of the lungs' air sacs, or alveoli. The oxygen molecules change from their state as a gas freely circulating in the air, dissolving into a solution in the plasma within the capillaries of the alveoli. Once in the solution of the blood, 98% of this dissolved oxygen is taken up by passing red cells, leaving just 2% remaining in the physical solution unattached. Red cells are particularly well suited to transporting oxygen because they contain a special oxygen-binding protein known as hemoglobin. Each molecule of hemoglobin itself contains 4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish blue in color and is called deoxyhemoglobin. It is heme which makes it possible for the red cells to pick up oxygen dissolved in the blood, transport it combined with hemoglobin, and release it back into the blood as oxygen in solution, ready for delivery to the various cells of the body. Hemoglobin gives up its oxygen as red cells travel through capillaries in tissues where there is a low content or partial pressure of oxygen. The partial pressure of oxygen represents the level of dissolved oxygen in plasma. As oxygen is released and again is carried in solution, the partial pressure of oxygen in the capillaries becomes greater than the partial pressure of oxygen in the surrounding tissues. This causes oxygen to move out of the capillaries into the tissues and to finally reach the cells. This graph, the oxygen dissociation curve of hemoglobin, shows why hemoglobin is particularly suited to its role in transporting oxygen. The oxygen dissociation curve demonstrates the relationship between the oxygen carried in combination with hemoglobin, the O2 saturation, and the partial pressure of oxygen in the arterial blood. The sharp upstroke and the flat plateau illustrate how oxygen is released to the tissues over a wide range of conditions. Its shape means that although the partial pressure of oxygen in the blood returning from the lungs and being pumped out by the arteries may be reduced to only 50% of the normal value, say due to lung disease or high altitude, hemoglobin will still be 85% saturated with oxygen.
Okay, hemoglobin is made of 4 globin molecules, 4 independent polypeptide chains, 2 alphas and 2 betas. It transports carbon dioxide through the aid of carbonic anhydrase, also nitric oxide, which is brought from the lungs to the tissues and induces smooth muscles to relax and lowers blood pressure. There are 4 heme molecules in hemoglobin that each contain a single iron atom whose job it is to transport oxygen. That's where the oxygen binding actually happens. Iron is critical for oxygen transport. It's absorbed in the upper small intestine. The absorption is increased by stomach acid and vitamin C. It's lost in the urine, in the feces, and in the menstrual fluid. So we need iron in our diet. Without it, we wouldn't be able to carry oxygen around the body. Other roles for iron include the electron transport chain, where it's vital in generating massive amounts of ATP in the mitochondria as a cofactor in that system of membrane-bound enzymes. Let's take a look at hemolysis and crenation now.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
Okay, erythropoiesis is the process of making the formed elements of blood. Red blood cells last 120 days in circulation. They lack a nucleus. Production of red blood cells, again, follows a very predictable sequence. The stem cells, which are in the red bone marrow, form pro-erythroblasts, which become early erythroblasts, which eventually become reticulocytes, and those become now red blood cells. Erythropoietin is the hormone that stimulates red cell production. It's made in the kidneys in response to low oxygen levels. This is one of the reasons why people that live at high elevations have higher hematocrits, because their erythropoietin production is stimulated by the low oxygen concentration in the air at these elevated levels. Okay, so you'll see in those individuals a higher red count, a higher crit than you would see in somebody that's lived most of their life at sea level. This is just a look at how erythropoiesis is stimulated. So we see decreased blood oxygen stimulating the kidneys to produce erythropoietin. This in turn stimulates the red marrow to crank out more RBCs, and this results in an increased oxygen carrying capacity in the blood. Okay, we want to end this portion of Chapter 19 with a discussion of hemoglobin breakdown. So take a listen, and when you're done, remember, study this material as it will be on the upcoming PRS quiz. And I'll see everybody in class. Thank you.
When old red blood cells rupture, the released hemoglobin is ingested by macrophages. The globin chains of hemoglobin are broken down to individual amino acids that are metabolized or used to build new proteins. Iron is released from the heme of hemoglobin. The remaining structure is converted to biliverdin, which is then converted into bilirubin. Iron is transported by transferrin in the blood to various tissues for storage, or to red bone marrow for making new hemoglobin. Free bilirubin is transported by albumin in the blood to the liver. Liver cells make conjugated bilirubin, which is excreted as part of the bile into the small intestine. Intestinal bacteria convert bilirubin into bilirubin derivatives, which contribute to the color of feces. Some of the bilirubin derivatives are absorbed into the blood and excreted from the kidneys in the urine.